Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is meant for 24th October 2020. We are into the revision mode. We have been revising chapter number 8, Human Health and Diseases, and we have completed the revision of the chapter in our previous session, and today I want to relate that with the experiment that is there in your class for your batch, that is exercise 14. Aim to identify common disease causing organisms and the symptoms of the diseases. Normally, how you perform this experiment, we keep a permanent slide, okay, or a specimen or a chart. You have to identify, write the identifiable characters. How do you say that particular slide which you are seeing under microscope is amoeba or paramecium, whatever it may be? So you need to first identify the organism. Second, you need to write the characters. And third, what are the diseases that it causes? So what are the symptoms of the disease? So this is normally, it's a 3-mark or 2-mark question in the practicals. Hence, so already I have told you how to write the experiment, what is the format in which you have to write. And if you recollect from your memory, I said the first will be aim. You have to write the aim of the experiment. Second will be principle involved. And the third will be the procedure. And I said the procedure should be in the point wise and past tense because you are doing it. So it becomes a past tense, point wise, past tense. After procedure, you are going to write observations. What is that you have observed? And observations also again point wise and past tense. After observations, fifth one will be conclusion. Based on your observations, what do you conclude? And your conclusion should give you result. And then finally, what precautions you should take. Since you are observing the specimen or slide under microscope, how should you handle the microscope? How should you focus the specimen? How should you focus the slide? How to use the focusing, magnification, okay, high power, low power, all these precautions you must write about it. And then after writing it, as you have observed in class 11th, in your biology record, there will be 2 types of pages. One is lined page, unlined page. In lined page, you will write all this. In unlined page, you have to draw and label the specimen you are seeing or the slide you are seeing, permanent slide, or the chart. So you need to write on the white page. So with this background, moving ahead, the first one in the format is aim to identify common disease causing organisms and the symptoms of the disease. This is your aim. And the principle involved, it is related to chapter 8, Human Health and Diseases. There are quite large number of organisms that are parasitic, pathogenic to humans. These organisms substantially damage the human body and cause various diseases, which may even be fatal sometimes. These organisms exhibit characteristic features in their external morphology. Symptoms of the disease caused by them are also specific. This is the principle involved. What are the required materials you need to have? You need to have permanent preserved specimens like we are having in our biology lab, permanent slides, we are having it, photographs, we are having it, and then of course you need to observe all this under compound microscope. And procedure, observe the preserved specimens or slides, photographs, and note down the features in the practical record book. Take care to observe all the minute details. This is important. When you are particularly observing under high power, all the minute details you have to observe. Draw labeled diagrams of the pathogens, disease causing organisms. So in that first one that you are likely to get, first slide is Entamoeba Hysteretica, or normally we say amoeba. So observe the following features of the parasite in the slide or photograph. Normally we give you permanent slide, okay? You have to focus because that involves a lot of skill. Under high power you have to focus. While focusing under high power, you have to take care of the distance between the object and the object lens. the gap will be so narrow. And already I have told you precaution that when you are focusing a slide, a specimen under the high power, you should not move the high resolution one, okay, or smooth and rough adjustment you should not do, only smooth adjustment you should do. So what do you see when you focus it under high power? You understand that entamoeba is unicellular. Shape of the cell is irregular due to the presence of pseudopodia. Pseudo means false, podia means foot, false foot. A single nucleus is present eccentrically in the cell. In the nucleus, a peripheral ring of granule of nucleoprotein and central karyozyme are observed. Rest of the space in the nucleus looks empty, like in figure 14.1. A few food vacuoles may be seen in the cytoplasm. Contractile vacuoles are absent. Mature quadrinucleated cysts may be present. Now, when you observe, it may not be all these 6 points that are listed here. Whatever you observe, that you must write, and you must write in the past tense. Draw the diagram and label it. Label it like this is plasma membrane or cell membrane. You can label this as cytoplasm. You can label this as food vacuole or vacuoles. You can label this as nucleus. You can label this as nuclear material. You label this as pseudopodia, PSEU, pseudopodia. Okay, this is how. And you must know what is the function of pseudopodia, what is the function of plasma membrane, what is the function of cytoplasm, what is the function of food vacuole, what is the function of nucleus, and what is the function of the nuclear material. So you must know every label, what is the significance of it, right? Next. Note, Entamoeba is an intestinal parasite. It is present in the intestine in humans and cause amoebic dysentery. The symptoms of the disease are loose motion, of course, mucus-filled water stools, abdominal pain and spasms. Of course, when it is attacking the children, it may lead to death also. That's why it is very, very dangerous disease. Systemic position, classification, it belongs to phylum Protozoa, class Rhizopida, type Entamoeba Hysteretica. That is, you can say biological name. At least this much you must write. Next, another slide we keep, that's Plasmodium vivax. I am sure you immediately, the moment you hear this name, you go for the Plasmodium cycle or Ronald Ross cycle. So it is intracellular endoparasite. Endo means inside. Seen easily within the RBC of the infected person. Remember, Plasmodium requires 2 hosts. Okay, one is the human being, another is the female Anopheles mosquito to complete the life cycle. Of course, it is unicellular. The most diagnostic stage of the parasite is signet ring stage in erythrocytes, within which it appears as a rounded body. It has big vacuole inside, and the cytoplasm is accumulated at one place containing the nucleus. Because of the above mentioned features, the parasite appears as a ring. Search the stage in the blood film slide, find the signet ring stage and draw it as a labeled diagram. So first draw the diagram and label it. It's a protozoan. Of course, all of us know Plasmodium is a protozoan parasite causing malaria in human beings. When an infected female Anopheles, see, this is female Anopheles carrier, mosquito bites a healthy person, it injects the infective stage sporozoite, you must remember this word, sporozoite, into the peripheral blood vessels. The infective stage undergoes several rounds of multiplication in liver as well as erythrocytes. So 2 target organs in us. One is the liver, another is the RBC. What are the symptoms when the malarial parasite attacks us? Intermittent high fever with the chills. That means intermittent means one day fever will be there, one day normal. One day fever will be there, one day normal. And that too high fever, something like 103, 104 degrees. So Fahrenheit. And chills followed by profuse sweating at an interval of alternate days. That is the symptoms that our body experiences. Classification, it belongs to protozoa, it belongs to class Sporozoa, and scientific name is Plasmodium vivax. And then the third one, of course this is not a slide, you must have observed in our biology lab. Specimens are there. Ascaris, that's a roundworm. The external features of roundworm are follows. Body is long, 20 to 40 centimeters. Of course, if you take scale, long scale, no, 30 centimeters, you can measure. And it is cylindrical, 5 to 6 millimeter diameter with no segmentation. Unlike in Annelida, earthworm, there will be segments. Here, no segmentation, no metamerization. Sexes are separate, as you are seeing, male and female. Female is lengthy, male is short. And both the females are longer than the males. Both the ends are pointed, as you are seeing. Both the ends are pointed. You can see anterior as well as posterior areas are pointed. Posterior end of male is curved. Look at this curvature in the male. You can see the curvature. And mouth is situated at the anterior end and is surrounded by 3 lips. One present mid-dorsally and rest 2 lips are situated ventrolaterally. For viewing these lips, a magnifying lens is required. Single longitudinal lines are present on the dorsal, ventral and on the 2 lateral sides, all along the length of the body. Out of these, the lateral lines are comparatively more distinct than other lines. You can see the vertical lines. Excretory pore is present on the ventral surface, slightly behind the anterior end. In addition to the ventrally curved posterior tip, the male worm has a pair of pineal spicules very close to the cloacal opening. In case of a female specimen, a female genital aperture is present mid-ventrally, about 1/3 distance from the anterior end. What about classification? Phylum Aschelminthes, class Nematoda, and the biological name Ascaris lumbricoides. And what of the danger? Roundworm or Ascaris is one of the common parasites found in the intestine of human beings. What will happen if it is present inside the human beings? Irregular bowels, so one day loose motion, one day constipation, so alternatively constipation, loose motion, occasional vomiting, and of course the most important thing is anemia, because it eats off the tissues and the hemoglobin content comes down. That results in this. The hemoglobin content comes down, anemia comes. And next one is ringworm, trichophyton, ringworm fungus. It is a fungus that feeds on keratin of the skin of human beings. The features as observed under the microscope are: texture of hypae is waxy, glabrous to cotton-like, cotton-like fibers. Unstained hypae are white, yellowish brown to reddish brown in color. The filaments are in that color. Systematic position, it belongs to kingdom fungi, Deuteromycetes, and the biological name is Trichophyton rubrum. What are the symptoms? Most of us have experienced this. Ringworm is a contagious fungal infection of the skin. Skin infection, it is basically infected area of the skin is itchy. Okay, itching sensation will be there. Red, raised, bulging, scaly patches with sharply defined edges. It is more red on the periphery than in the center, creating a ring-like appearance. So this is a fungal disease and very, very difficult to go. So these are the related things in the experiment, and you need to maintain a recording all this whatever we have discussed now. So keep on maintaining the record.